Great. Hey, as everyone is joining us, delighted to welcome you all to Club EvMed. My name is Vaughn Cooper. I'm at the University of Pittsburgh in the School of Medicine and the director of our Center for Evolutionary Biology and Medicine. And I'm glad to do the introductions today on behalf of the Club EvMed team. So welcome to Club EvMed Conversations. Our goal is to connect the evolutionary medicine community and share exciting new research. We like to have this as informal as possible with an emphasis on discussion and presentation of different perspectives, which is why we're calling it Club EvMed Conversations. Club EvMed is organized by the International Society for Evolution Medicine and Public Health. It now is a great time to become a member or renew your membership. Please consider supporting the society by submitting articles to the society's journal, Evolution, Medicine, and Public Health. There are many editors online right now, like myself. Club Med is also organized by 10 evolutionary medicine centers and programs around the world. And welcome especially to our newest collaborators, the Brazilian Evolution and Health Study Group, located at the University of Sao Paulo, the University of Kiel, Kiel Evolution Center, Transevo, and the Evolutionary Studies Group at Vanderbilt University. So our process, please do participate in the conversation, post questions and ideas in the chat box during the presentation, which sets the stage for discussion that follows. And I will try to moderate that. Please raise your hand virtually to ask a question anytime. We'll call on you after the presentation, and please feel free to use your social media platforms to talk about Club EvMed. So with that introduction, I'm delighted to introduce today's speaker, Dr. Áine O'Toole. She is a postdoctoral researcher at the University of Edinburgh, studying the evolutionary process of emerging viruses. And she's also developed a number of bioinformatic tools for genomic epidemiology. On a personal note, I got to learn about her contributions, which were significant during COVID-19. And I'm delighted that the research that she's presenting is about something that I stumbled into myself, along with others, which is that the genome signatures of our pathogens show signatures of our own immune system and our own defenses in their nucleotides. That is, the mutation spectrum of the viruses are affected by our own immune system. So Eve has carried out a master's by research at the Smurfit Institute of Genetics in Trinity College Dublin. She got her PhD in the Wellcome Trust Host Pathogens and Global Health Program, and since really contributed to the field by software tools like Pangolin and working in the phylogenetics core of the COVID-19 Genomics UK Consortium and as part of the Arctic Network. She also served as chair of the Pango Lineage Designation Committee, which is clearly perhaps a thankless but necessary service to the community, and has been involved in teaching genomic research viral genomic workshops around the world. She's continued in the Rambo lab at Edinburgh, working on the evolution of emerging viruses, including mpox and poliovirus. And so today, again, the talk is APOBEC deaminase editing and mpox virus is evidence for sustained human transmission, since at least 2016. Eve, take it away.
Okay, thank you so much. I'm going to try share my screen, which worked earlier, so hopefully it'll work again. And okay, perfect. So firstly, I just wanted to say thank you again so much for the invitation to speak, and also thank you for everybody who's tuned in today to listen as well, but also to speak. So feel free to jump in if anything's not clear, or if you have questions or anything like that, and then also hopefully there'll be plenty of time at the end as well, and we can have a chat. Sort of broadly, what I'm going to talk about today is sort of setting the scene for what was the original MPXV paradigm. So what how we came to know MPXV as a zoonotic virus. Talk about the current outbreak, and I'm saying current because there are still ongoing cases of this outbreak to date. And there are potentially other unlinked cases that are also cropping up now as well, but current outbreak, but obviously the one that started in Nigeria a few years ago. And then some of the evolutionary implications and possibly public health implications as well for the work that I'm going to present. So first of all, a bit of a historical perspective on MPXV. So the very first human case of the virus was was detected in the DRC in 1970, and within a year, several other countries were reporting cases of the disease as well. It's historically been regarded as a zoonotic disease, with low numbers of cases occurring from spillover from the reservoir rodent hosts. Most of the cases that from early studies in the DRC, most cases were observed to be in children under the age of 10 and majoritively in young boys, so associated with exposure risks. The vast majority of cases were linked directly with exposure to the rodent reservoir, with small numbers, so the remainder usually associated with household contacts and really quite rare onward transmissions detected. Over the following few decades, there were quite a few small outbreaks that occurred across West and Central Africa, and also notably one in the USA as well. I think, interestingly, in 2005, epidemiological investigations into an outbreak in what is now South Sudan revealed 5 serial transmission events. So although onward human transmission is rare, we've known that it's certainly possible for some time. So if we look at the diversity of the virus, the genetic diversity of the virus, broadly it splits into 2 major clades, which have some geographical linkage, but the true ranges really are not particularly clear. So to sum up, the kind of MPXV paradigm up until recently was that it is a zoonotic virus that causes a disease because of exposure to local rodent hosts. The disease occurs usually primarily in young children with really limited capacity for onward transmission. And if we fast forward to 2017, a number of MPXV cases were reported in Nigeria. At the time, suspected cases were pretty widespread, but the majority of confirmed cases were in the southern states. And sequencing at the time, genome sequencing at the time, revealed that most of those, all of those cases lay within the diversity of clade 2, but they had significant enough difference from one another that it was not ruled that it was a single outbreak, because the paradigm at the time was that it's a zoonotic disease, and the fact that there was some diversity there appeared that that these cases were not linked. And a few small numbers of cases were reported over the subsequent years, with a few international cases, small numbers, but they were linked directly to transport from Nigeria, so to travel from Nigeria. And then in 2020, there was a very small number of cases reported. However, it's unclear whether this is as a result of lockdowns or whether it's just a reporting issue because of the SARS-CoV-2 disruption. But by 2021, the cases were back up to what they'd previously been. What was different about these cases being reported was that they were mostly in adults, which is obviously different from what we'd previously seen, and also mostly in males as well. So mostly adult males between 20 and 50 years of age. So clearly quite different. And then by 2022, there was quite a few cases reported. Now, this increase in cases doesn't necessarily mean that there's a large outbreak occurring. This could be some sort of sweep because of increased exposure to the rodent reservoir. However, a lot of these cases were reported in urban centres and didn't have any obvious link to any sort of rodent reservoir. So, and given the different demographics of the cases as well, there's clearly something different about this outbreak. So between the 13th and 26th of May 2022, 23 nations that hadn't previously reported cases of the virus had began to report this. And then I think we all are fairly aware of what happened after that. To date, so I think this is the latest figure from the WHO. There's been nearly 94,000 cases in 117 nations. So the outbreak then sort of swept across the globe. If we look at the early genome sequences that were shared, again, they were linked to clade 2, which is similar to the sequences that had already been produced. But what was interesting, these are some early genomes produced from a Portuguese group. They linked to some of the earlier sequences linked to the Nigerian outbreak over the years, but are linked by this really long branch of 42 unique SNPs. And for a double-stranded DNA virus, this is really quite a significant amount of mutations. We'd expect about one nucleotide change every 3 years. So seeing this amount of changes in just a few short years is really quite a striking thing. So as more genomes started to pile in, this section of clade 2b got renamed, a section of clade 2 got renamed clade 2b. And if we look just at clade 2b here, so we're just zooming right into the cases that were reported in Nigeria, with a slightly earlier sequence that's also part of the clade from 1971 in Nigeria. You can see immediately that there is quite a lot of genetic diversity here. So what we're looking at, the pink triangle represents the major global lineage that's spread throughout the world. And we've got some of the earlier sequences that have been, some earlier cases that have been reported and sequenced either at the time or also retrospectively when the outbreak gained traction. So we can see here that branch that was pointed out earlier of 42 SNPs. So one of the retrospective sequences that came out from a group in the US from 2021 breaks up that diversity, but you can see that this is quite a large number of SNPs. And just for the purposes of communication, but also for this talk, I'm just pointing out that there's a nomenclature system implemented for these MPXV lineage, for MPXV, proposed by Happy et al. And for this talk, I guess just the key thing to remember is B.1 is the global lineage that that kind of came out of Nigeria and then circulated in majoritively the MSM community networks. And the other A lineages are lineages that are majoritively circulating in Nigeria. And in most of the results I'm going to show today, we take a single representative sequence of the B.1 lineage. Because it was so richly sampled and sequenced, for the purposes of our analysis, we don't want to bias it by including way more sequences from that exact lineage. So unless I say specifically otherwise, we're just using a single B.1 genome in most of our analysis. So if we look at that phylogeny and take a look at the mutations that occurred throughout it, so this is the clade 2b phylogeny, including the Nigerian sequences and only a single B.1 sequence, we can see there's this incredible skew of G to A and C to T mutations. And when you see some sort of skew like this, you think there's something immediately, there's got to be some sort of unusual evolutionary process going on here. And again, from I suppose from my PI's experience, working with HIV through the years, and knowledge of various host immune editing processes, we were able to identify that this potential skew is consistent with APOBEC3 deamination. So I'm going to convince you, but just to give a slight explanation of APOBEC3. So it's a cytidine deaminase that binds to single-stranded DNA and deaminates cytosine to uracil. And then that gets synthesized across onto the negative strand and also copied back onto the positive strand again. So when you observe this mutation, we actually see a C to T mutation. And then crucially, APOBEC3 has a strong preference for binding TC dinucleotides. So what we actually observe is a TC to TT dinucleotide mutation. And then, of course, this can happen on the positive and the negative strand. So when we're looking for signatures of APOBEC3 editing, we're looking for TC to TT and GA to AA mutations. So if we go back to our plot here, so here we're seeing G to A and C to T, but if we color this by whether those mutations lie in the context of APOBEC that APOBEC3 prefers, you can see that the vast majority of these mutations are consistent with APOBEC3 editing. Now, when you compare that to the other two MPXV clades, you've got clade 1 and clade 2A, you can see that it's really striking, the comparison. You don't see this skew, and you don't see this enrichment for APOBEC3 editing, APOBEC3 preference. And if we put numbers on it, it's 90% of observed mutations versus 13 and 8%. So it's really quite a strong signature. And remember, the numbers that we're looking at up here, I'm not sure if you can see my cursor, but hopefully you can. The numbers that we're seeing up here, that does not include the B.1 lineage. It's only got a single representative. So you're looking at the ancestral, the diversity ancestral to B.1. But if we only now look within the B.1 lineage, which we know is transmitted from humans to human because we've observed it, we can see that same extreme skew. And actually, there's even more extreme a skew in the diversity prior to the B.1 lineage. So we see this enrichment for APOBEC3 type mutations. So to recap, the current outbreak has gone global. We know that it's different. It's got different demographics to previous. There's clear, consistent human-to-human transmission, and the mutations that we observe within this outbreak seem to be majoritively consistent with APOBEC3 editing. And so you can ask that question, why are we suddenly seeing APOBEC3 acting in the MPXV population? And the clue might actually lie in the host genomes. So APOBEC3 is a family of cytidine deaminases, and mammals have 3 ancestral paralogs of this. Because of a gene fusion event and a gene loss event, rodents only have a single APOBEC3 gene, whereas humans and other primates actually have 7 copies because of duplication events. So it's possible there's additional functionality within humans, or that the virus may be more exposed to APOBEC3 editing when it's in a human host rather than in a rodent host. So what does this actually mean for MPXV? What are the evolutionary consequences and public health implications of the fact that the virus is clearly the evolution is being driven by this mutational process? So the thing to remember about APOBEC3 is that it's primarily antiviral in function. and most mutations are deleterious in general. Also, the APOBEC3 mutation repertoire is really quite limited. It only has the capacity to introduce TC to TT mutations and GA to AA mutations. And it's also not reversible by the same mechanism. So any mutations that are introduced at this higher rate can't be reversed by APOBEC3 and can only be removed from the population at a lower rate. So talking about most mutations being deleterious, if we look at the distribution of mutations that we observe across this phylogeny, if we just look at the APOBEC3 mutations, we can eyeball it and say, okay, there's some non-synonymous, some synonymous, there's a kind of distribution. We see some intergenic mutations as well. But if we look at that in the context of the TC or GA target site availability, so of those targets that APOBEC3 could edit, how many of them are being edited, if you get me. So for example, here you can see the proportion of non-synonymous mutations that we observe is a lot lower than the availability, the proportion of available non-synonymous ones. And we see the synonymous ones are slightly enriched. But obviously you can kind of eyeball this, but putting some numbers on it, the probability of getting this many synonymous mutations, given the underlying distribution, is actually really very, very low. So what it does seem is that we're observing the residual least harmful mutations by APOBEC3 that after natural selection may have removed the more deleterious ones from the virus population. Oh yeah, so we are. So we see this in the survival bias in the genome. Looking at the mutation repertoire, so the fact that it has this really limited repertoire for inducing TC to TT mutations, what that really means is that only 15 amino acid substitutions are possible, and 3 of which produce stop codons. So here, what we're looking at is what the observed amino acid distributions are across all APOBEC3 target sites in the genome, and then that's on the left-hand panel, and then on the right-hand side, this is the hypothesized amino acid that would be induced if APOBEC3 edited that target site. So you can see there's quite a large portion of synonymous, so that it doesn't induce any amino acid change, but really it's quite a small number. And if we color this by what we predict is how big an impact that mutation would have on a protein. So this is a bit of a fudge, because what we've done is categorized amino acid changes by Grantham score, which just essentially ranks amino acids and how different they are. So based on things like polarity and size and that sort of thing. So here we're just coloring by whether or not a change induces a significant amino acid substitution. And you can see there is quite a few synonymous, but there is potential for inducing some quite radical changes. And then keep bearing in mind that these can be induced in a single direction and can't be removed by the same mechanism. What we're seeing here is that we've got a kind of this system and this mutational process that can induce radical changes into the virus. So because of all this, you can ask the question if it will produce a longer term loss in virus fitness. What that length of time is possibly something that could be modeled, but we haven't done it. But that's for another day. Another implication of the fact that there's this increased generation of genetic diversity because of the fact that APOBEC3 is editing a double-stranded DNA virus, which otherwise would have quite limited diversity, is that we can take these mutations generated and use them to analyze and do genomic epidemiology on this outbreak. So what we've done here is taken the phylogeny and we've reconstructed all of the mutations that have occurred across this. So we've color-coded all of the different mutations by whether they're consistent with APOBEC3 editing or not. And we can see this root branch. So this is where we assume somewhere along this branch, the virus emerged into the human population. What we can do is take a APOBEC3 root to tip and construct a sort of APOBEC3 clock. So by doing this, we're reconstructing the most common recent ancestor of this phylogeny. So here at the base, and comparing the number of mutations of APOBEC3-like mutations from the base to through each tip. We do that for every tip across the tree, and that's what this plot is. So along the y-axis, we're seeing the count of APOBEC3 type mutations from root to tip, and then along the x-axis, we're looking at the date of sample collection. And you can see that APOBEC3 mutations do accumulate at a fairly linear rate. There's quite a fair bit of noise, but it's definitely a linear relationship. And they're accumulating at about 6 mutations per year. We can do, so you can see here, that's the single B.1 representative that we've included in that phylogeny. And we can do the exact same thing just within B.1. So this is using that the 2021 sequence from Maryland as an outgroup here. So we're using that as the most recent common ancestor between that and the B.1 lineage as the root, and doing the same thing. And if we plot that against on the same plot, we can, even though obviously the time frame is a lot shorter, we can see that the mutations are also accumulating at about 6 per year. So it's consistent. And then what we can do is say that if we believe that when APOBEC3 edits began accumulating is when MPXV emerged into the human population, we can extrapolate that linear relationship back down to when y equals to 0. So when there were no observed APOBEC3 type mutations in the phylogeny, and say, okay, this could be the time of emergence. Now, obviously, this is an incredibly simplistic way of doing it, but this gives an estimate of the 20th of June 2015. So that's quite a simple way of doing it. However, we obviously don't know that every observed APOBEC3-like mutation is actually because of APOBEC3. There could be a small number that may not be. They could have come about through just normal evolutionary processes. So if we suggested that 1, 2, or 3 of those APOBEC3-like mutations are due to just other reasons and not due to APOBEC3, this pushes the estimate forward linearly in time. So depending on whether you look at 0, 1, 2, or 3 APOBEC3 mutations being attributed not to APOBEC3 editing, it could be between the 20th of June 2015 or mid-December 2015. So of course, this is still quite a simplistic way of doing it, but we're not incorporating any sort of uncertainty into our estimate. And we're also not using the information that's provided by the non-APOBEC3 mutations either. So we wanted to try a different approach, and we wanted to try and attempt to model this using BEAST. So if you're not familiar, BEAST is a software package that uses Bayesian inference to estimate time trees. And you can use it because it's quite flexible. You can use it to test various evolutionary hypotheses and also estimate various different parameters like the time of emergence, which is what we wanted to use it for. So we developed a model where we were using an epoch model that was really quite straightforward. This is where you have two defined time periods, and the first defined time period was that there's just no APOBEC3 acting. That we're assuming in the rodent host reservoir, we don't have APOBEC3 editing taking place in the MPX. Our MPXV is not affected measurably by APOBEC3. And then in the second epoch, after emergence into the human population, we see APOBEC3 acting. So the way we kind of did this in brief is that we took the MPXV alignment as our input data, and this is a very, very zoomed in portion of it. And we identified the sections that are APOBEC3 targets and non-APOBEC3 targets. So we had two different sets of data, and then we were going to treat them differently. So the non-APOBEC3 data, we just let evolve under standard substitution model, whereas the APOBEC3 data set, we allowed only two states, so it can be edited and not edited, and we also only allowed mutations go in the forward direction as well. And then we let this sort of background rate evolve across the whole tree, whereas we only enacted the APOBEC3 rate in our second epoch. So some of the results out of this, one of the really striking ones is what we'd expect is that the APOBEC3 driven evolution is 28 times faster than the background. So what that means is that when you're looking at this current outbreak, we're looking at this really quite elevated rate of evolution. So comparing what's happening within that versus what's happening in the other clades, you have to bear in mind that an equivalent branch length within the rodent reservoir could be hundreds or even thousands of years of evolution, whereas what we're observing within this outbreak is a different process. Another output of the analysis produced an MCC time tree, so the most probable tree, and what we're looking at here are the same phylogeny as before, but now the tips are placed on time, so when the date was sampled, and then all of the internal nodes are estimated using BEAST, and we just haven't included the outgroup sequence for clarity here, so that we can zoom right in. So the two of the parameters that were estimated were the time of most recent common ancestor, which is the when all of the samples that were taken coalesced, and then before that, sometime before that, the time of emergence that was estimated based on our analysis. And we can see the distributions here. So with the model, we estimate that the time of emergence of APOBEC of MPXV into the human population is most likely September 2015, but with bounds that extend between August 2014 and July 2016. So what we can say from that analysis is that the transition to sustained human-to-human transmission occurred at least as early as July 2016, but probably earlier than that. So it had been circulating in the human population for quite some time before the global emergence in 2022. Another estimate, another output that we can get from this model is a sky grid, which essentially just estimates the population size of the virus population at different time points through time. And here we can kind of see that it is obviously increasing over time. But if we compare this to an exponential growth model, the fact that this completely overlaps implies that the MPXV population has been exponentially growing since its emergence in 2016, despite in 2020, where there are far fewer cases reported in Nigeria. So despite the lower case numbers, there doesn't seem to be any genetic evidence of the population growth not continuing as it had been before. So just to recap the sort of take homes, with our beast analysis, we can estimate that the emergence of MPXV into the human population occurred at least as early as July 2016. The fact that we have this APOBEC3 driven evolution has implications for the long-term viability of the virus in the population, as well as our ability to detect human outbreaks going forward as well. And the fact that we're now observing this sustained human-to-human transmission means that when we're observing new outbreaks of MPXV, this is something we need to be considering, and we definitely need to revise this zoonotic paradigm that we had previously had. So I wanted to thank all of my co-authors on the paper, and the senior authors here at the bottom are indicated as well. Thanks to all of the institutions involved. A particular thanks to the Nigerian CDC, who have incredibly detailed situation reports, so have meticulously detailed every part of the outbreak since 2017 when the first cases were reported. Peter Borges et al., who kicked off and shared the first MPXV genome sequences from the global outbreak, which then kicked off this kind of open data sharing of MPXV genomes. Also the CDC and DSTL UKHSA for genome sequencing and generation and sharing as well. And also to my PI, Andrew, for all of the help with BEAST too. So thank you very much for listening. And if you have any questions, please ask. I'll stop sharing.
Thanks very much. The floor is open to questions. And I don't see any in the chat just yet. Please raise your hand if you're interested.
I'll start things off.
Right on, Charlie.
Okay. Fantastic talk. That was so nice to see and to have this walkthrough of all the amazing things in the paper you guys published last fall. Thank you so much. I wonder if you could say more about the duplications of APOBEC3. I think you said there's 7 copies of it in humans. Is that true across primates? Do primates tend to have multiple copies? And is anything known about that and why there's so many copies?
So yes, to the primates. Primates also have 7 copies. So it's something that occurred prior to that split. Why that is the case, I'm not sure. I know that they all likely have slightly different functions, and since this paper has come out, there's been wet lab studies that have actually shown that it's APOBEC3F that is editing MPXV in vitro. So it does seem that this one specific APOBEC3 that's inducing these mutations, whether that's because of where the virus localizes, or whether it's something along the lines of a kind of dosage or transcription difference. I did try and look at the rodent APOBEC3 because there's a single functional copy, and it is definitely still expressed as well, and is upregulated during infection. So there's some publicly available transcription data for mice that shows that that is the case. So, but I don't have any more information about why that isn't actually affecting MPXV. Pox viruses have a kind of reputation for putting a lot of their genome into host immune defense mechanisms. So I think some studies have looked and seen that up to 40% of the genome of pox viruses can be purely to try and defend against the host immune system. So clearly, clearly, human host immune system is doing a better job of it than the rodent is. So, or no, or yeah, we'll see. Yeah.
Yeah, Jamie.
Okay.
Hi, thank you for a really wonderful talk and for the great work. I'm curious if you've tried any of this on clade 1 viruses in light of the reported big increase in clade 1 cases in the DRC in the last year or so.
Yes, but I'm not able to comment on that. I'm sorry. We have, and it should be out soon.
I was wondering if what you've thought about, like, well, I'm sure you've thought about how this signature can be used to more broadly extend dating in new zoonoses. Have you, so I guess, and it's a broad question.
That's the same question, but I can answer that one. Yeah.
Yeah. I mean, just like, just sort of, very broadly, and maybe even for other viruses.
Yes, and I think that's, so I can't say for other viruses necessarily, but I think for within MPXV, there's nothing to suggest that we wouldn't see the same pattern in other outbreaks. And I think it's something that I think is quite important now for trying to estimate how long outbreaks have been going on. So, so the fact that it's accumulating 28 times faster, we're able to observe enough APOBEC3 mutations after a relatively short amount of time to get a sort of temporal signal. And also trying to, without the APOBEC3 mutations, it's really quite difficult to rule out just a sweep of a rodent reservoir. So this is what I think is really quite great about this as a signature, because say, for instance, you had a large sweep, it was all fairly similar in the human population, and even if epi information suggested that it was a human-to-human transmitted outbreak, it would be difficult to rule out that there was a sweep of an outbreak in the reservoir with multiple spillover events, because an outbreak in the reservoir is going to look very similar to an outbreak in the human population in the absence of APOBEC3 mutations. So I think it will be quite useful going forward to help rule in or out whether it's being human-to-human transmitted. But obviously we need to wait a certain amount of time before you're going to be able to actually detect the APOBEC3 editing.
Yeah, I mean, just
it's not that fast.
To sort of echo that point, we noticed what we thought was were signatures of APOBEC in, once we sort of got to the first, say, 100,000 SARS-CoV-2 genomes, and we mentioned something, you know, about that in this preprint that remains orphaned, but we didn't really know what we were doing. So I'm delighted to see this inference being sort of formalized. So yeah, thanks. Katja, good to see you, and your floor is yours.
Yeah, that was great. I really enjoyed that. I'm wondering, do you know when during infection these APOBEC3 mutations actually kind of arise? Do you have any longitudinal data from infections? Do you see them over the course of infection or like shortly following a transmission event?
I personally have no idea, but I think that would be really quite interesting. I know the way pox viruses replicate is this sort of rolling circle amplification. in the cytoplasm, right? So presumably at that point when single-stranded DNA is exposed, because APOBEC3 binds to single-stranded DNA. So it must be during replication. And
Yeah,
yeah,
I was thinking
yeah.
more along the line of an acute infection, if
Oh,
there's
within an
anything,
infection.
deleterious mutation to the virus, right? Presumably.
Yeah.
And so you wouldn't
Yeah,
actually
yeah.
see an increase in frequency, right, throughout the course of an infection, right? So I just wonder whether it has to do with kind of very small bottlenecks, right? And the kind of very, early on, very early on replication dynamics that have that deleterious mutation fixed in the viral population shortly after transmission.
Yeah, so I know in Edinburgh so I'm not personally involved with this study, but I know the hospital that has access to all of the samples. The outbreak in Edinburgh, they have so every case that came in, they've sequenced at least a couple of times. So they do have this longitudinal data. I'm not sure what the actual output of that is or not yet. I think it's potentially still an ongoing analysis, but that data does exist, and I'd be really interested to see if you see these mutations coming up. It could be that they're not competitive, that that maybe you'd have to maybe sequence quite deeply if you were doing that. They may not rise in the virus population, but it'd be interesting to see for sure.
Yeah, thanks. Appreciate it.
One of the things, actually, I don't know, I think this is probably related. One of the things we noticed with the B.1 outbreak was that we saw convergent mutations cropping up a number of times, which suggests that there is quite a lot of APOBEC3 editing going on, and that the fact that in such a short span of time you're already seeing the same mutation arise multiple times suggests that that we are observing just a subset of the mutations that have come about. Yep.
I had a quick
go ahead,
question.
Johnny.
Yeah, a quick question in the chat, just kind of broadly asking what's your hypothesis or yes, your assumption on any reverse zoonoses likelihood? And have you seen any evidence for it?
In MPXV or
sorry.
just
Yes.
yes. Yeah. So I think the very first case of MPXV was in a monkey in Denmark, which is why it was called monkeypox. I'm not sure whether that was a case like a human actually transmitted back to a primate or not. A lot of the sampled cases are in rodent reservoir hosts, which wouldn't, that's just regular. I'm not sure if anyone else here knows better than I do about whether there's been sequences from or samples taken from pets or anything like that. I'm not sure. Not that I've heard of. Yeah.
I was wondering about
yeah.
domestic pets or things like
yeah, that's the first thing that kind of popped into my head. I haven't heard anything of that now.
Okay.
But I suppose if the rodent reservoir, I'm not sure, how infectious it is for cats or dogs.
No, that's okay. Thanks. Yeah, I was just kind of
See if
broadly
anyone
asking.
has a pet rat, we could ask them. Yeah.
Any further questions? Well, it looks like we're all tuned in. I don't want to shut down any further discussion, but this has been really enlightening. I mean, I'm delighted to see it and really appreciate you joining us in Club of Med to share your work, Eve.
No, thanks so much for having me again and for listening and the questions and everything.
Absolutely. Johnny, do you have anything else to add?
I wanted, hopefully this is not overstepping, but if you have questions, I feel like maybe Áine, you could take some emails later afterwards.
Absolutely. Yeah.
Yeah, because we post these online and people often ask me a lot of things and I just kind of forward them off. So if that is okay, we will do that. Yeah.
I have one
sure.
quick thing to add. I was just racking my brain in my hard drive. There is one case report of a human to dog transmission of mpox. And I'll throw it in the chat right now. It was just kind of a one-off. But
do you know if that was recently?
It was back reported in 2022, and it was people sleeping with their dog, I think, like just allowing it in the bed, and so they were wondering about fomites and sheets and things like that.
Was it
but-
sequenced?
Hey, good question. I don't see it here. PCR confirmed. Oh, wait, sequences were obtained. Yeah, okay. So
Okay.
look for it. The link is there.
Cool.
But it's just one generation in a dog. So as far as that takes you.
Not, yeah, not much. Not unless they monitored the kennel. Well, anyway, thanks again.
Hey, thank you, Áine. That was fantastic.
Yeah, Anya, thank you so much.
Great to see everybody.
Bye. Thank you.
Bye, everyone.
Bye now.
Bye.